It's great to be here again and present to you some of these findings that we have from some of the mahi and the research we've been doing in the region. It's likening it to looking at a health check of the lake. This is a term that colleagues in our scientific discipline have used before, that if you go to the doctor now with an illness, say, I've got this pain in my chest, the first thing he asks is, have you had that before? What's your history? Do you know if somebody else in your family has had it? Or have you experienced this in the past? So you might go and start monitoring your lake now and look at it all going on, and you can see that it's got this and it's got that and it's got this other thing wrong with it. But unless you really know its history, you're not sure if that's been there in the past all its life or that it's a recent phenomena. And that's what we use these mud cores for, these sediment cores, to go back in time, in history, to understand the health of the system of the lake. And the mahi that is done, as I said, consists of some of the sort of traditional approaches, the water quality samples that are taken, and here's the co-lead of the project, Susie Wood, with one of our students taking water quality samples from the lake. We look at what's trapped in the sediment, in the mud at the very top of or at the bottom of the lake I should say, but where the lake and the water meet, because that's representative of the recent changes that have happened in the lake. So the water tells us what's going on right now. The top bit of the mud that's being scraped off into a container tells us what's happened very recently. And then we take these larger mud cores to look at that history. We're trying to capture 1,000 years of time in these mud cores. The reason for that is that it provides us usually with the opportunity to get back before humans arrived in Aotearoa, before Māori first arrived in that time frame in large numbers anyway, and it gives us a sort of a reference and a benchmark for what the natural variability of that lake has been, what it's been doing, and how it's responded to different changes through time. First with the arrival and settlement of Māori in different regions, and then of course the arrival and settlement of European and their activities.
I'm just thinking about Ōnoke, and since the Ruamahanga was diverted and didn't go into Wairarapa Moana, that has meant that any silt, sediment or whatever is being brought quite readily straight into Ōnoke, which means that there would be a whole lot of sediment in the lake that is all very recent.
Potentially, yep.
So that means that you'd have to possibly take a deeper core sample there than somewhere else that didn't have that same issue. Is that right?
It can be that way. It depends on how much actually gets flushed straight out versus into the rest of Ōnoke as well.
So there's a channel through
there are channels, or there has been.
A sample from the base of the channel as well as somewhere else?
We wouldn't necessarily go in the channel because that also gets scoured out as well with the larger flows coming through. So we will probably, for the scientific cause, we'll try over towards the other side of Ōnoke where it's a little more settled. With these cores, we aim to get 1,000 years. That we don't always get that. Some of them, and I'll point them out later, because of a large event, whether it's floods or things like that, they fill in very quickly. So a large flood event could take up this much in a core, and then we don't get back 1,000 years. But others we do. But we still capture some element of their history. Things that we'll talk about today, the team will talk about today, is just a little bit of the work we've done on Lake Wairarapa. We'll talk about the current lake health as sort of shown by the water quality and some of the sediments that we collect from the very bottom of the lake, and Sean will talk about that and some of the ideas around sediment nutrients, the nutrients that are trapped in that mud. I'll then talk about some of the catchment change that has occurred in the history of the lake, and we use things like pollen, little pollen grains. These are microscopic images of them that are trapped in the mud. And that tells us how the vegetation has changed native forest to any sort of disturbance that might have happened with people or setting up farmland and so on. We also look at the mud itself, so going from a clay or a mud to a fine sand as an idea of erosion in the landscape. We also look at things like algae and bacteria in the lake. So some things I'll present on are these little remains of microalgae, diatoms, these little shapes here. And we also use some new techniques looking at the DNA trapped in the mud, so the environmental DNA of bacteria and specific bacteria that are indicating lake health. So some bacteria you probably hear about things like cyanobacteria, and often cyanobacteria is related to toxic algae and algae that cause sickness in humans or sickness in animals, but even just bacteria that are usually associated with causing these algal or bacterial blooms, the big green blooms that you see on some lakes. So what's their history? What have they been doing? When we look at the core and talk about these changes, I'm going to break the time frames up into pre-human and then sort of the evidence of Māori settlement that we identify through some of these different indicators, particularly the pollen, and then we look at the sort of post-European time frame. And now go back in time and show you lots of squiggly lines with different colours. And now I'll try and explain the squiggly lines with different colours. So you have to remember, put it all against the mud core, with the top being now, the bottom being sometime in the past, and we can talk about how far that is. But we know from certain things in there where we are in time, and that's where we sort of talk on the far right there about the post-European, the Māori settlement sort of period, and then what we look at is pre-human. So in any of these little slides I'm going to show you, if the wiggly line is down close to the main line here, there's not much of whatever I'm talking about. If it jumps up, there's more of it, okay? So less, more, and that's the same all the way along. So for example, the charcoal here, when we look at charcoal as an indicator of, while there's natural fires in the landscape, we look at it as an indication of human activity in the landscape. Whether it's small clearings or large-scale clearings, whether it's on purpose or accidental, we use that as indications of that. So through this part of time, there was not a lot. There's still natural burning going on, bits of charcoal coming into the lake and from the catchment. But then at a period in time, there's a large amount of charcoal reflected there. The charcoal and the bracken fern pollen are two markers that we use to indicate the sort of presence and persistence of humans in the landscape, whether it's Māori or European. You know that if you clear away a little bit of vegetation or a slope here, the bracken fern will be one of the first things that comes back in and populates that landscape. And the gorse, exactly, when gorse arrived, but gorse has only been here since the European, whereas bracken is native and it's been in the landscape that whole time. So it's a colonising plant, and it fixes up, sort of aerates and increases the fertility of the soil.
What is bracken?
It can actually be quite a tough, coarse fern, and it grows up to, it can be this high, but most of the time you'll see it's around this height. Hard to get through. But yeah, it dominates after fire in particular. It's lovely and green when it first grows, and then it goes really woody after that. So the fire and the bracken are normally associated with each other. You get fire, you get the bracken fern, and we use that as an indicator of humans and the environment. Other things we can see here changing through time. So clearly the fire and the bracken were up there during human arrival. But associated with those changes then, this is looking at the size of the mud and the sand and things in the core that we take. So there was very fine silts and clays through most of its time frame here in Lake Wairarapa before humans arrived. And then after humans arrived, we do see these increases in the size of the sands and the muds in the lake. And again, that's indicative of more erosion. So if you start to burn the landscape, clear away the bigger trees, get the bracken fern going, the erosion comes with that. And this one curve on the side here is pollen from the manuka that was growing around the catchment through time. So manuka from this pollen record is likely to be quite abundant, particularly most likely around the margins of the lake, as we still see it today in a number of places. Probably quite dense thickets and stands. That was quite abundant around the lake, but then it was a consequence of the firing and some of the clearing that those manuka stands become less abundant until we got what we have up here today. It's always still around there now, but not as abundant as it used to be. Now, one thing that is sort of superimposed on top of this that I'll just mention, but I won't sort of talk too much about it, is that the Wairarapa, Wellington, of course, is a very earthquake-prone location as well. Some of these changes where we see in the past where there's been pulses of coarse material might be results of previous earthquakes that shake the mountains, shake the rimatakas, the landslides that occur and slowly feed naturally into the lake itself. So that's what some of these historical shifts might be. We also know, of course, through time that these earthquakes have happened where humans have been here too. And some of the signals we see up through the top part of the core are likely to be a mix of landscape erosion from human activity, but also probably pulses of earthquakes that have occurred through that time as well. So it's a bit of a mixed signal between the naturally occurring events and then the human ones. This one is just more an expanded version of what we were looking at. So the mud core, the sediment core, tells us about the forest that was around as well. And that, again, can be a wee bit of doom and gloom because it shows what was there and what may not be there now. But it also highlights some of the things that were there that in restoration that you might want to try and look at getting back to and having more abundant around the landscape. Back in time up till now, so we have some of the key ones such as the rimu, the kahikatea, mira matai, totras, and of course the beech forest that grows around in the higher hills as well and down on the lowlands, and of course some of the things like the manuka. The grasses are generally a sign of, again, if you disturb the landscape you'll get native grasses coming in, but then closer to the top that's the type of grasses we get with the farming coming in. And of course the bracken fern is a big key one that jumps up there, and actually I mean that in some cases too there were a lot of the catchment again being shaken around a bit by earthquakes and so on. We do get a lot of tree ferns that, you know, the punga tree ferns and things that we see in the landscape, they were relatively abundant as well. The sediment core can tell us about some of those changes too, about the types of plants that were there. And some of those may now be, you know, seen as taonga species for different reasons. And I know Raz talked a lot about the manuka and the properties of the manuka for filtering things out. Well, we can see that those plants were there in reasonable abundance in the past, but may not be there now. Leading on then, we can look at what's going on in the lake. And the next two slides are around some of those things about the algae and the microalgae that we were talking about. You just need to get an idea that some of these things were all the way through there and they've changed through time. But key things here is some of these things live on the bottom of the lake, on the mud. Some of them attach themselves, the little algae, those diatoms attach themselves to plants. So the macrophyte plants that we were talking about, if there were more of them in there, there'd be habitats for different things. Some of them indicate that the lake was more turbulent in the past or not. And then some of them are sort of free swimming in the water. But this one in particular is usually associated with poor water quality in the lake system, and then some of them are indicative of more river input into the lake through time as well, or drainage coming out of the catchment. So we can see that there's a few different changes here. There's always, even though there's small changes, you know, some of the ones here, there were more of these diatoms and algae that are natural in the lake that used to attach themselves to plants, but in more recent times as the plants have gone away that live in the lake, those have become less abundant, and we see sort of rises in some of these ones that are just free swimming in the lake and becoming associated a little more with the turbulence of the water, and then again that sort of mixed up lake system. As I highlighted there is this one that is indicative of poorer water quality, and we see that more now right at the top of the mud core, indicating recent times, most likely to be associated with the sort of European time frame there, but it was never present in the past. So again, that you can sort of even hazard to go, well, if we can get back to a point somewhere down in here, at least that water quality's, there's a reference marker for going back in time, as Ra was talking about. And we see the influence of some of these more river types of diatom and algae coming in and then disappearing, and that may also interact with the changing of the Wānanga River going in and out of that lake, but also changes coming out of the catchment.
So are the diatoms baddies?
No, they're not. Some of them are associated with different environments, and that's what we use them. They're algae that are always living in the lake. Some of them can cause, diatoms can cause algal blooms, big green horrible mats and things like that, or in the rivers as well. Not all of them are baddies, some of them are fine. They're all part of the ecosystem because things live on these. So, you know, your little aquatic invertebrates and things that are in your lake, that your fish, that your tuna and kura feed on, they feed on this stuff.
How can you tell what the lake should be,
Yep.
or if you could say that it should be?
Yeah, sure. So you look at key time frames and you kind of get an amalgamation of all of these different things, I suppose. All these different types of lines of evidence you can pull together to say this is what we think that ecosystem was like at the time. So, you know, as Ra was saying, it might have had this type of aquatic plant, it might have had these types of trees growing around the margin, and we'll get onto some other things, and it might have had this type of bacteria composition, or none of the toxic cyanobacteria. You know, that's what we think it should have been in its most common reference state. But I will also say is what it was then, or what you might say it should have been, might not actually align with your values that you have now. You might actually want it to be a little different, or you may say that's actually really hard to achieve, but we could get something in between and it looks like this. You know, you're sort of painting a picture using these different lines of evidence.
So Marcus, I've been using the kind of bacteria markers, so if they're lower or not as prominent, then I've been thinking that's a healthier waterway. Is that a mistake?
It's one indicator, and I think that's probably something that that we, in bringing all of these different lines of evidence together, are trying to get away from. It's a good start, don't get me wrong. If your lake has got cyanobacteria in it, toxic algae or bloom-forming bacteria, and that's been present there for the last 40 years, then you probably don't want that state and whatever's associated with it, nutrients and things. But that is just one measure of water quality usually, and it doesn't often incorporate the rest of the ecosystem that we're trying to build as well, because it's just one thing.
Should we build out of what we've got here an indicator of ecosystem health?
I think so, and that's why I was really excited by the cultural monitoring indices, and I love that idea moving forward, is that you'll bring in a bunch of different things, whether it's taonga species, and particularly you've got a lot of your tuna, your kora and other things already in there, but what about the plant life? What about your manuka as a taonga species, because it's an important part of this ecosystem? What about the little things that we don't know much about, the diatoms, and or even things like the little water fleas that eat the diatoms? It's to keep them under check, or that eat the algae. Those are all part of an ecosystem health model that I would look at. And that's sort of like, again, that reference to the body. You're looking at everything. You're not just looking at your sore arm or your stomach contents, you're looking at everything.
Yes, we have done some work done on some of that type of approach.
Yeah.
And others.
Yes, sorry about overbearing questions, but in terms of the DNA, are we getting a little more genome sequencing out of the DNA?
Yep. So we are, I'll come on to some of that data now, we can talk a little bit about that. So this is the world of DNA, but bacteria environment. So the DNA, you know what DNA is in your body, it's in every little cell you've got, and if you die or whatever, that DNA can be preserved in the mud too. Usually it breaks apart, but often we can look at what's left of the DNA strands within the mud. So we're looking at one of the successful things that the colleagues, Susie Wood and others and colleagues at Cawthron have been able to do is look at the DNA of the bacteria. And bacteria are everywhere, you know the bacteria, but there's lots and lots of them. So you can imagine that it's actually like getting a whole bunch of information in your hand and going, what am I looking at? But you can also then pick out key ones that we're interested in. For example, all these coloured bars, this is some of the key different types of bacteriophylla that are at this depth in the mud core. And then at this depth you've got all these other different ones, and at this depth you've got all these other different ones. So what we have to do now, and you can see this is the pre-human and post-human time frame, and there are some colour changes going on in there. But we have to filter out now the key ones we're actually interested in that are related to water quality, lake health and things like that. Now, so for example, and I'll just go through this and then I'll address Ra's question a bit. We can see that some of the key bacteria that are in the water in the pre-human time frame are associated with more salty water conditions, saline conditions. And that makes sense because we know from the geological record and from some of the prehistory work that's been done that, you know, the Wairarapa, Onoki, while they're connected but semi-separate now, they were much more open and almost as a continual bay in the geological past. So this sort of thing makes sense. As we come through time and get into the out of that sort of more saline environment coming into the sort of human era, we see that there's these dark blue bacteria that start to increase over time, and these are some of the bacteria that are associated with some of the what Sean was talking about, the bacteria that start to utilise the nutrients that are in the mud or in the water and increase the algal and bacterial productivity of the lake. So they're starting to, they've always been present, they're always there naturally, but as we come through time and come into, say, the European zone, when we start to get more fertilisers or erosion coming in, these bacteria start to increase. And then right at the top, in the top sort of layers, where we had the diatoms that were showing not so good water quality, we also now get this cyanobacteria, which is often associated with the algal blooms or the toxic algal blooms. So that's only a recent arrival in this lake. The DNA technology is increasing month by month on what we can do with it, about all the cool things we can pull out of it and what species in detail we can get to. So while we're focused initially a lot on the bacteria, but we can also now look at the DNA of the different macrophyte plants and go, actually, we can tell that this species of plant was here and this one wasn't here. It's not always telling us about the abundance, but it can give us a good indicator of what was around.
How about different species of Vandergoes?
That work is being done. So they're doing that with, they can definitely do the gene sequencing on the Manuka. But I don't know, we've yet to figure out whether we can pull that apart from a mud core. But all of those are sort of future horizon possibilities, but that stuff's moving so fast now that when I say future horizons, I always think of maybe 5 years, but it could be in a year or two's time for all we know. But this also coming back to, where might you try and have a goal of bringing the lake back to, if you can try and do some planting, stop the nutrients coming in, stop the sediment coming in. You can try and look back in time to go, where in the scheme of things would we try and get the lake to a condition that it's healthy. It's probably never going back to the saline conditions because it won't be an open bay anymore. Onoki may still have this sort of signal in it quite prominent, but where could we bring Wairarapa and some of the other lakes into its healthy ecosystem? So this is just really a little bit of a summary, I suppose. This knowledge, this history, this kōrero from the lake, it's another knowledge to weave into all the mahi that you are doing. It gives you some reference points to move forward to. It gives you the ability to weave in as I say, the knowledge on the mātauranga of the movement of people in the region. The practices that you did around Hingakai can be woven into this. And I think, again, I know there's a lot of yearning for solutions. And I'm not necessarily saying that this work isn't always going to provide the magic bullet, the solution, but it certainly does provide a lot of stepping stones in the right direction, or pieces of the puzzle that you can choose to take to try and guide restoration and prioritise what to protect in the lakes in the region. We focused on Wairarapa. We've done the same sort of work from Lake Ngānoki. We've also been working on Lake Panui, the two lakes at the Pinkau area. And the other lakes we'll be looking at, lakes and lagoons as they're classified. Over the next 2 days we'll be sampling those in the region as well. So some of these have been connected, we know, in the past with Lake Wairarapa, or they're very much influenced by it. Like Alton Perry from the Regional Council has talked about that these two lagoons in particular, they're influenced by the height of Lake Wairarapa, so when Wairarapa's high, they're high, and so on and so forth, so they can get quite a bit of change. These other ones are a little more disconnected in some ways. Rototawa in particular is a small round lake. We don't know how deep it is, but it's nestled in behind some of the old dunes that were formed in the past around the lake. This is just a big acknowledgement to all our partners of the project, including, starting with the iwi that we've connected with all throughout Aotearoa, and the role that they've played in this project, and then our regional council partners and so on. So as the space shuttle came over New Zealand, it took a photo, and the brightest thing shining is Lake Wairarapa. Thank you.
Thank you.